All right, here's part 2 of chapter 12, still focusing on the adaptive immune response. And this is where we left off with part 1, looking at how the T helper cells, part of the cellular branch, helps to activate the cytotoxic T cells, CD8 positive T cells, also part of the cellular branch. And T helper cells, CD4 positive T cells, will help the B cells, the humoral branch. Within the T cells, all right, the different T cell effector cells, there are 4 major groups. All right, here you have the activated T helper cell, CD4 positive, and with the different cytokines that it is able to secrete, those can differentiate the T helper cells into 3 different types: T helper 1, T helper 2, and T regulatory cells. Now, why is there a difference? Well, the difference is the specific effector functions of these cells. So T helper 1 cells will produce certain kinds of cytokines, and those cytokines actually activate cytotoxic T cells, the CD8 positive T cells. And then these cells will go on and do their cellular response by killing those infected cells. T helper 2 cells will secrete a different batch of cytokines. So if I would have made this picture, I would have made these circles here, different color. All right, because say maybe the orange ones come from T helper 1, but maybe red ones or blue ones come from T helper 2 cells. They are different. The cytokines from T helper 2 cells stimulate B cells. And so then the B cells can then become either memory cells or plasma cells. And plasma cells, if you remember, secrete antibody. All right, the immunoglobulin that is not stuck to the surface of the B cell, but rather is secreted. And then T regulatory cells, the T regulatory cells actually decrease an immune response once that threat has passed. And so it regulates the T cell response to basically say, okay, the problem's over, so go home and get some rest type thing, instead of continuing to do the same effector job. All right, with cytotoxic T cells, you want the cytotoxic cells to kill the virus-infected cells, but then once the virus-infected cells are dead, you don't want them to continue to kill good, healthy cells. All right, so for that to happen, the T regulatory cells come in and down-regulate that response. If the T regulatory cells are not working properly, then you don't have a properly maintained immune response. Now, there's another topic within immunity, or within the immune response, and that is tolerance, or sometimes referred to as self-tolerance. All right, so B cells and T cells, when they are being made and activated, either in the thymus for the T cells or the bone marrow for the B cells, all right, they're screened for self-tolerance. Can they tolerate the self? If they can't tolerate themselves, then they're going to attack it. And so they're screened for self-tolerance to avoid self-attack. So in this case, do the T cells recognize the MHC, which we haven't talked about yet, but we will. And if it's yes, then it's potentially self-reactive, and then it goes on. If it is self-reactive, the cell gets the signals to undergo apoptosis or cell death. If it's not, all right, at this point, it does not recognize self-antigen in the spleen, or in the lymph node, then it passes self-tolerance screening and migrates to additional lymphoid tissues and can become activated. All right, same here. All right, with the B cell, could it make antibodies to self-antigens? If the answer is yes, it immediately or is supposed to immediately undergo apoptosis because you don't want antibodies made to your own tissue, your own proteins. Otherwise, it will start attacking whatever that protein is. Could it make antibodies to self-antigens? No. And so it passes that screening for self-tolerance and migrates to the lymphoid tissues, the lymph nodes, the spleen, to further go through activation. So a good compare and contrast of T cells and B cells. All right, adaptive immunity mediated T cells mediates the cellular branch, B cells the humoral branch. And what cells do these include? T cells are T helper cells, CD4 positive, and the cytotoxic T cells. All right, recall that within the T helper cells there are the T helper 1, T helper 2, and there are also T regulatory cells. Site of maturation is the thymus, primarily found in the lymphatic tissue. Remember, very low levels in circulation. Same with the B cells over here. Antigen recognition receptor, T cell receptor, B cell receptor for B cells. Requires antigen presenting cell to become activated. Yes, the T cell needs its antigen processed. And that is through the antigen presenting cell, which we looked at on one of the earlier figures. Major histocompatibility complex. We'll talk about this in class. T cells respond to MHC class 1. MHC class 1 is found on all cells except for red blood cells. And considered an antigen presenting cell? No. B cells, humoral branch, plasma cells are the effector functions. And requires antigen presenting cell to become activated? No. It needs the T cell for the cytokines, and memory cells made after activation, yes. Major histocompatibility, MHC class 1 and class 2. All right, that is what it expresses. And the class 2 is because it is a MHC or an antigen presenting cell. Considered an antigen presenting cell? Yes, because it has MHC class 2. All right, so MHC class 1 and class 2. We will look at this in class, all right, but MHC class 1 interacts with CD8 cells on cytotoxic T cells and CD4 on helper T cells, and antigens presented with MHC class 1, it's going to be intracellular. With MHC class 2, extracellular. All right, here's a picture of what's going on. MHC class 1, all right, that is going to be able to interact specifically with the cytotoxic T cells. So this is going to be CD8, all right, along with the T cell receptor. CD8 right here, T cell receptor, you have the antigen here. MHC class 2 is going to specifically interact with the B cell receptor. Or sorry, this is T helper 2, T helper cells that express CD4. All right, this is the T cell receptor, CD4. Both of these molecules must interact in order for there to be positive selection of that cell. And this is the antigen presenting cell. All right, the professional antigen presenting cells are dendritic cells, which is what this guy is. It is also macrophages, and it is also B cells. So B cells express MHC class 2, and so B cells can actually interact with T helper cells and allow for activation of that helper cell. All right, then there are a particular group of antigens that are classified as super antigens. All right, not because they are superheroes, they actually cause a lot of problem. All right, but with super antigens, or with regular antigens, remember they sit right in that little antigen binding site. They are very specific. And with this, you have that little triangle. Here you've got this notch that the bottom of this antigen fits into. And each T cell receptor, each B cell receptor is going to be very specific for its own antigen right? Whether it's this knob-like one, the triangle, whatever this guy is, it's very, very specific. So when you have this antigen that comes into the body and it gets attached to this antigen-presenting cell, all right, maybe only 4 out of the million B cells or T cells can actually interact here and get activated. And so then you have a small immune response, but it's very specific to this antigen. With superantigens, there's a very different way that it binds. All right, and so we'll just go to this figure here. The superantigen binds to the outside of the T cell receptor and also to the MHC molecule here. And so then instead of just activating this one T cell because it was specifically interacting in this antigen binding site, you activate 100,000 T cells, all right, because it's a super antigen. It binds outside of the antigen binding site, but it's still able to bring the antigen presenting cell and the T cell together, and it gets activated. And so it's not processed at all. It directly binds to the MHC and the T cell receptor to cause what's considered a bulk nonspecific activation. So large numbers of T helper cells are activated, and this causes dangerous levels of cytokines to be produced. And this leads to toxic shock syndrome. You guys may have heard of that. And it's toxic shock syndrome because so many T cells are being activated, the level of cytokines is massive, and it eventually causes the body to go into shock. And then, unfortunately, that can lead to death. Staphylococcus toxic shock syndrome is a big one, and that really is the major one. There's also a streptococcal made by strep species of exotoxins, but typically superantigens are going to be produced by gram-positive bacteria. All right, activated T cells and B cells, it's showing just T cells here, will undergo proliferation and differentiation. All right, the proliferation is just an increase in number. And here you have the interaction between the antigen presenting cell, its specific antigen, CD8, interacting with class 2 MHC, class 1 MHC here, and the T helper cell interacting with the MHC class 2 with its antigen and the CD4 co-receptor, and that produces cytokines. All right, and this T cell is producing cytokines that activate this T cell, but will also cause the dendritic cell to become activated, and then these cells proliferate, and so you have high numbers. All right, then this process of producing cytokines can also then cause the cell to differentiate, to change from just a general T helper cell to T helper 1, T helper 2, to the memory T cell, and then likewise the memory cytotoxic T cell, or the cytotoxic T cell that's going to go out and actually seek and destroy infected cells. All right, so after proliferation and differentiation, there are the effector functions that can start. Remember, T helper 1 can stimulate cellular responses, so it's going to activate the cytotoxic T cells. It can activate the natural killer cells and the macrophages, and then all of these cells can go and do their own function. T helper 2 will stimulate the humoral immune response to cause those B cells to differentiate themselves into antibody-producing plasma cells. All right, and this is where we will stop this is where we will start for part 3.